Hello, I'm Dr. W. John Martin. This is the third of a series of new videos to remove any doubts regarding the existence of stealth-adapted viruses. The first video described the use of low stringency polymerase chain reaction to look for virus-related sequences in patients with the chronic fatigue syndrome. Positive results were seen in over a third of the tested patients and in some patients with more severe brain damage. Cerebrospinal fluid and a brain biopsy were also positive, even though there was no inflammation. The sequences being detected in some of the viruses appeared to include both Herpesvirus sequences and a retrovirus sequence. The second video described the culture of atypical viruses from two patients. The viruses were unusual in the type of cell damage or cytopathic effect. It comprised cells rounding up and developing a foamy, vacuolated appearance in the cytoplasm. I invited Dr. Paul Cheney to come and look at the cultures. Dr. Cheney, along with Dr. Dan Peterson, had observed the outbreak of chronic fatigue syndrome in patients living near Lake Tahoe in Nevada. This video will discuss the visit by Dr. Cheney and some aftermath. Dr. Cheney visited my laboratory in Los Angeles in March of 1991. I showed him the strongly positive virus culture and the foamy vacuolated cytopathic effect. He also saw the patient who was still in a coma from whom one of the viruses was isolated. This was a patient in the Los Angeles County Hospital with a 4-year prior history of bipolar psychosis. I think he was impressed by what he saw. Dr. Cheney had relocated from the Lake Tahoe area to Charlotte, North Carolina, I think around the end of 1986. He was being supported in his efforts to understand chronic fatigue syndrome by Mr. Marc Iverson, a retired banker. Mr. Iverson had formed the CFIDS Association of America, CFIDS referring to chronic fatigue immune dysfunction syndrome. Part of the support that Dr. Cheney was receiving was being provided for collaborative experiments with Dr. Elaine DeFreitas, a virologist working at the Wistar Institute. I knew of this collaboration because in September in 1990, Dr. Cheney and Dr. DeFreitas jointly announced at a Japanese conference that they had identified a retrovirus in chronic fatigue syndrome patients. The retrovirus was closely related to human T-lymphotropic virus type 2. Dr. Cheney told me that Dr. DeFreitas had prepared a publication on which he was a co-author. Other co-authors included Dr. David Bell from Lindenville, New York. Dr. Bell had identified an outbreak of chronic fatigue syndrome among children. The other co-author was Dr. Hilary Koprowski, the director of the Wistar Institute. The paper was to be published in the Proceedings of the National Academy of Sciences. This shows the title of the subsequently published paper. Dr. Cheney recommended that I apply to Mr. Iverson for financial support. Mr. Iverson and Dr. Cheney kindly agreed to divert some of the funding going to Dr. Cheney to my laboratory. Mr. Iverson wanted my research to be consistent with that of Dr. DeFreitas. He therefore asked me to emphasize the polymerase chain reaction data indicating a relationship to a retrovirus and not to mention the possibility of an altered herpesvirus. I went along with it because I knew that at least certain genes in some retroviruses could induce a foamy vacuolated cells. Specifically, Spumavirus is a type of retrovirus that causes foamy vacuolated cells, the word spuma being the Latin word for foam. Now, simple retroviruses have only 3 coding genes. They're referred to as GAG, or group antigen, that codes for the capsid protein, POL, which codes for the polymerase enzyme, and N, that codes for the envelope protein. This set of 3 coding genes is flanked on either side by non-coding long terminal repeats, or LTR. Now, the Spumavirus has an additional gene complex located between the envelope and the right-sided long terminal repeat. It is therefore called the Bell gene. Now, it was known that the Bell gene in and of itself was able to induce foamy vacuolated cells in the brains of infected transgenic mice. I therefore envision that the Bell gene or something similar could have been inserted into herpesviruses, other viruses, or even into other retroviruses and induced the cytopathic effect that I was seeing in the cultures. Now, Mr. Iverson was also in discussions with Mr. Ed Taylor, a pioneer in the early satellite industry. Mr. Taylor had the resources to substantially assist the CFIDS Association, I think primarily to promote the work of Dr. DeFreitas and Dr. Cheney. My research was presented to, or at least interpreted by Mr. Taylor as being in direct support of the work of Dr. DeFreitas. I was taken off guard and really disappointed when Mr. Taylor called me to inform me that he had notified the Centers for Disease Control that I had actually isolated a Spumavirus. He argued that my work had directly confirmed the retrovirus theory of Dr. DeFreitas. It would have been far better had he simply said I had cultured a virus that caused foamy cells. Now, Mr. Iverson also informed me that he was growing concerned that the nationwide growth of the CFIDS Association was being challenged by Dr. J. Levy of the University of California, San Francisco. Dr. Levy did not believe that a virus was causing chronic fatigue syndrome. Rather, he thought it was an overactive immune system. To competitively assert the interest of the CFIDS Association, I was asked to give a talk at a conference being organized in San Francisco on September the 4th of 1991 that would be attended by Dr. Levy. My talk was going to be followed by a press release that would mention the possibility of Spumavirus. Mr. Iverson would help ensure that the press release would be published by Newsweek, the New York Times, and the Wall Street Journal. I was also scheduled to attend a conference being organized by the Centers for Disease Control for the 19th of September. The major theme was going to be a discussion of Dr. DeFreitas' research. Although Dr. DeFreitas didn't attend in person, she gave a lengthy presentation over the phone. And I also was required to mention something about our own research. I had difficulties with both conferences. Although I had interesting observations based on the polymerase chain reaction and based on viral cultures and electron microscopy, I still did not have actual DNA or RNA sequence data to truly characterize a virus. I also felt awkward being linked into the retrovirus camp and with Dr. DeFreitas. I don't think she fully understood the polymerase chain reaction and had not included all of the relevant data in her paper. I had also problems with the political and the competitive overtones attached to the CFIDS funding. I recall one particular clinician talking to Mr. Taylor. He insisted upon calling him Dr. Taylor because he knew so much about the illness. It seemed to me that he was ingratiating himself in an effort to receive funding. I was also somewhat surprised by how little clinical interest there was in following the individual patients. Dr. Jay Goldstein was essentially ignored at the San Francisco conference, and pretty much the same occurred to Dr. Cheney at the CDC. Nevertheless, I was justifiably being criticized for the press release orchestrated by the CFIDS Association. I had tried to publish my research and had sent an article to the Lancet, to New England Journal of Medicine, and to the Journal of the American Medical Association. I don't think anywhere in the article did I mention the word Spumavirus. Each of the journals rejected my article. I had another setback when Mr. Walter Gunn, who had recently retired from the Centers for Disease Control, sent me some blood samples. Samples came from 10 adult patients selected by Dr. Paul Cheney and 15 adolescent patients selected by Dr. David Bell, along with some controls. Dr. Gunn announced at a conference held in Albany, New York, in October of 1992 that I had identified 58% of the patients as being positive and 50% of the controls as being positive. This terminated any financial arrangement or support I had from the CFIDS Association. I would have thought that my suggesting the possibility that the patients selected as the control patients might have a 50-50 chance of being infected with some unknown type of virus would have created some interest. It did not. Rather, it added to the criticism that I should not even be studying chronic fatigue syndrome, and certainly not at a major university. Now, in the next video, we'll return back to the science and provide more optimistic hard data based on the DNA sequence data obtained on the cultured viruses. Thank you.